Well, welcome everyone who has arrived to our industry partner update with RegenX Bio. We're joined by Vivian and Naz. I'll just give a quick overview for anyone who is new to Defeat Duchenne. We're the country's only national charity dedicated to ending Duchenne muscular dystrophy. Our goal is to provide leadership in research, advocacy, and support to ensure every Canadian affected by Duchenne can live a long, active life. And we look towards a future without Duchenne. So I'd like to welcome you to this webinar with information from RegenX Bio. They look forward to answering your questions. I want to mention that there is a Q&A button. Please use that for asking any questions. The chat is not as reliably looked to. So if you have a question, the Q&A function is the best way to get the attention of speakers. I'm happy to introduce you to Vivian Fernandez, who's the Executive Director of Patient Advocacy for RegenX Bio. And at that, I'm going to stop sharing my screen and hand it over to Vivian to introduce.
Okay, Naz, and who's gonna advance the slides? Okay.
Yep, I'll share the slides here.
Okay, perfect. Hi,
Great.
everybody. I'm Vivian Fernandez. I am the Head of Patient Advocacy here at RegenX Bio, and I want to thank Judy and Defeat Duchenne Canada for organizing this webinar. Naz and I have been looking forward to this call for quite some time, so we do appreciate the opportunity we have today to connect with families in Canada to introduce RegenX Bio and RGX-202, which is our investigational gene therapy for Duchenne. We'll talk about AFFINITY Duchenne, our ongoing clinical trial. We will go over the data that we've shared so far publicly from the phase 1/2 portion of AFFINITY Duchenne, and we'll introduce the design of the phase 3 portion of the trial, which is active, open, and enrolling patients. And I just want to say that we are super excited to be expanding this very important program into Canada. This is a program that is near and dear and extremely important to all of us at RegenX Bio, and we are extremely committed to our work in Duchenne and to helping bring better treatments for Duchenne muscular dystrophy. And just super excited to be part of a vibrant and very engaged community, a role model for other rare diseases for sure. Our hope for a better future for those that are affected or impacted with Duchenne motivates us. And your trust in us, in what we do and what we say, is what drives us to work harder and smarter. So if you could go to the next slide. These are our forward-looking statements. Go to the next slide. So who is RegenX Bio? We are an AAV gene therapy company founded back in 2009. Our headquarters is in Rockville, Maryland. For those of you who may not be familiar with the geography in the US, we're just in the northern suburbs of Washington, D.C. And we have now about 350 employees working here at RegenX in the US. Our mission is to improve the lives through the curative potential of gene therapy. We are leaders in gene therapy. We are developing gene therapies for rare disorders as well as what we call more common disorders. Thousands of patients have been treated with approved and investigational products that are built on our proprietary NAV technology platform, which is our library of AAV vectors. And we have a number of products in late-stage development. Also know that we have the expertise and the resources in-house to develop gene therapies from a preclinical stage through clinical trials and beyond that. And we also have a state-of-the-art manufacturing facility right on our site at our headquarters. You can go to the next slide. This is our pipeline, the products that we have in development. And as you can see, we work in two key areas. rare diseases, which is up top in blue, and retinal disorders in orange. And with the retina disorders, we actually deliver the therapies directly to cells in the retina. In rare diseases, we have programs for MPS types 1 and 2 and Duchenne. In our retina program, it includes programs in wet age-related macular degeneration and diabetic retinopathy. All of our programs are in late stage, so either advancing to phase 3 or in phase 3 clinical trials. And it's an exciting time of year for us because we actually have our first product in with the U.S. Food and Drug Administration in review for accelerated approval. This is our gene therapy for MPS II Hunter syndrome, and we're very excited about that, and we hope to have news in the months to come. And Duchenne continues to be our lead program for the company, and we're very grateful to be a part of this community. Go to the next slide. I'll stop here and I'll turn it over to Naz, who will introduce RGX-202 and walk you through our work in Duchenne.
Yeah, thanks, Vivian. So I'm going to talk to you about RGX-202, which is our drug to treat Duchenne. So RGX-202 is an investigational AAV or adeno-associated virus gene therapy that's designed to address the lack of functional dystrophin. So as you may or may not know, AAV gene therapy utilizes something called vectors. There are different types of vectors that are specific to get into different parts of the body. The vector that we are using is called AAV8, and this is designed to deliver the transgene through a one-time IV infusion to skeletal and heart muscles. What then happens is that the transgene itself has a portion called attached to it called the promoter. Our promoter is known as SPC512. That drives the expression of the transgene to allow it to make protein in the muscle tissue. And the microdystrophin transgene is unique to RGX-202. It's a synthetic transgene. It's a novel protein that's designed to retain the key elements of the dystrophin gene. So this is a designed transgene that's made to mimic what dystrophin looks like and the key elements that we think are important to include in here, because the full-length dystrophin cannot be packaged into the vector as it is too big. So you pick out key elements, mostly based off of really high-functioning Becker patients that have been historically described, picking out exactly what exons are included and what parts of the protein might be most essential to having this truncated or shortened microdystrophin function as closely as possible to naturally occurring dystrophin. And that's how you can see how the different companies have different types of transgenes that they're delivering. So our transgene is unique in that our promoter is a smaller promoter, so it allows for more packaging of the gene, of the transgene into the vector. And our transgene includes the C-terminal domain. So RGX-202 is the only microdystrophin gene therapy with the functional elements of the C-terminal domain. So you can see our drug here at the top compared to the other drugs that are available, I guess not approved yet in Canada, but on a research basis, potentially in the future in Canada for, I think, or I'm not sure if you have an ongoing trial in Canada with Sarepta's drug Elevidys. And then there's Genethon and Solid Biosciences. So you can see that between the companies, there are different vectors used, different promoters, and different elements of the dystrophin protein that are included. But key elements of ours are that we use AAV8 and we have the C-terminal domain. So why does the C-terminal domain matter? So we found preclinically, meaning in mouse studies on the MDX mice, that the C-terminal domain in the RGX-202 microdystrophin helps preserve muscle health and enabled muscle resilience. So it significantly enhanced restoration of the dystrophin-associated protein complex. I'm going to go back to this slide here. The dystrophin-associated protein complex is this little gray pocket of proteins here that lives in the cell membrane or sarcolemma. And it stabilizes the microdystrophin in muscle cells and potentially allows it to accumulate at higher levels and act for extended periods of time. The CT domain in RGX-202 microdystrophin also protected against contraction-induced damage, enabling better muscle recovery. So as families with children with Duchenne, you may know that eccentric muscle contractions, which means you're contracting against an extended muscle, like if you were to do a bicep curl or run down a set of stairs, for example, or walk down a set of stairs even. You have a muscle that's extended and you're contracting against it. So we did this test of eccentric muscle contractions in the MDX mice, and we found that after 20 muscle contractions, which is at the end point here at the x-axis, the amount of generated muscle force improved with the addition of the C-terminal domain as opposed to the transgene without the C-terminal domain. So here in gray, you could see how the DMD mice performed. So after 20 contractions, they could only produce 20% of the force. In a control mouse with no Duchenne, they could generate around 65 to 70% of the force. Without the C-terminal domain and the transgene, our mice could perform at around 40%, but with the C-terminal domain, they were able to generate closer to 60% of the force. So this preclinical or mouse study supported the decision to include this unique portion of our transgene in the RGX-202 drug. So now I'm going to go through interim phase 1/2 data. So a phase 1/2 study, as you may or may not know, is one that's set to really focus on the safety of the drug and administering the drug for the first time to children. And we are essentially fully enrolled in the phase 1/2 study at this point. And the way that it was conducted was first dose escalation and expansion. So we started at a lower dose, which is the lowest effective dose, and then we moved to a higher dose. We had to establish safety with the lowest effective dose. And our first dose was 1 × 10^14 genome copies per kilogram, and now we moved to 2 × 10^14 genomes copies per kilogram. We first started actually treating kids who were 4 to less than 12, and then expanded to meet the needs, particularly in the United States where newborn screening was coming up and running, to address treating kids who were 1 to less than 4 years of age as well. So by the time this trial completed, we dosed boys 1 to less than 12 years at screening who had genetically confirmed Duchenne muscular dystrophy with mutations in exons 18 and above. The reason why exons 1 through 17 were excluded to start was because before our study got initiated, there were prior gene therapy studies in Duchenne that had some serious adverse events that were thought to be potentially related to some immunogenicity, some regions of the exons in Duchenne that might trigger an immune response, and hence the serious adverse events that maybe lived in the 1 to 17 region and have since been narrowed down even further. We'll talk about that with the phase 3 design. So the kids in this study had genetically confirmed Duchenne with mutations in exons 18 and above. They had no pre-existing antibodies to the gene therapy, which is the AAV8 capsid. And for the children who were 1 to less than 4 years of age, they needed to be able to complete a 10-meter walk without assistance. So just walk 10 meters without any assistance. They could be on or off corticosteroids prior to start, and then had to stay on or off corticosteroids after, and needed a weight of greater than 10 kilograms. The 10-kilogram weight is mainly because we have to check a lot of blood work for safety reasons, particularly in the phase 1/2 study, and there's only so much blood that you can take out of a child who is less than 10 kilos, so we kind of capped it at 10 kilos. For those kids who were over 4 years of age, they needed to be able to complete the 100-meter walk without assistance and be on a stable dose of corticosteroids for 12 weeks before their baseline assessments. Now, what we do prior to dosing is take a proactive approach to preventing adverse events and serious adverse events in our study, which is a proactive immune modulation regimen. So what you may read is these regimens are a bit different between different companies. And our regimen is unique in that we designed it to be proactive again for improved safety outcomes. So we use something called eculizumab, which is a complement inhibitor. Complement is something that increases naturally in response to any kind of when your body senses the need to mount an immune response, possibly to a viral offending agent or fungal or bacterial infection. So this C5 inhibitor is a reversible C5 inhibitor, meaning that once it gets out of your system, it's not going to continue to inhibit. There's not long-lasting effects. And what we do is we give this to bind the excess complement that forms as a response to getting your viral load as part of the RGX-202 gene therapy. Every gene therapy delivers a viral load. We give this to you for about 5 doses. And we give this in combination with something else called sirolimus, which is also a T-cell inhibitor. And this is this is an oral medication. Eculizumab is an IV medication. The sirolimus is meant to help prevent not just an acute inflammatory response to the vector or the AAV8 that's being delivered, but also prevent something called an anti-transgene response, which could be almost similar to transplant rejection in some patients. And actually, sirolimus is a drug that's used for transplants, like bone marrow transplants or organ transplants. So you give that for a period of time, for 2 months, and then you give that in addition to prednisone. So you get this kind of big regimen for 2 months, and then you wean off of it in the 3rd month, and then you feel good in the sense that you've done everything possible to prevent anything. We'd rather be proactive about preventing anything from happening rather than playing catch up and trying to deal with an adverse event that happened that might be a very prolonged course of therapy and with a lot of sequelae or issues that come up afterwards. So thank goodness this has been a good protocol for us, and we're very happy with the regimen. And we find that it has the potential to reduce reactive safety interventions, which may or may not be successful at times, which we've unfortunately heard of recently. The phase 1/2 trial endpoints, so primarily, like I said, phase 1/2 is safety. The secondary endpoint was microdystrophin expression. So microdystrophin is the protein that's created by the RGX-202 drug, and we look at the expression of that protein within muscle tissue. And we also secondarily to that look at muscle function, and that includes the time to stand, the 10-meter walk run, the time to climb, the North Star Ambulatory Assessment for children who are over 4 years of age, and the Peabody Developmental Motor Scale for kids who are 1 to less than 4 of age. So I'm going to review some of the demographics and safety data from our phase 1/2 study and biomarkers. So I won't go through the full details of this slide, but what I want you to focus on is that we have dosed with 2 drugs. Again, dose level 1, which was our lowest effective dose, and dose level 2, which is our dose that we tested first for safety, and now we're going to be testing for efficacy. We've had a variety of patients that we've dosed, including children who are 3 kids who are 1 to 3 years of age and 7 kids who are 4 to 11. The average age that we dosed was a bit older than what you've heard in other studies. In the 4 to 11-year-old age group, it was 8.7 years old, with our oldest child being dosed at 12.1. And the weight has gone up to 35.5 kilograms, and we've had up to 18 months of follow-up that we're reporting on today. And they had a variety of baseline function, just to kind of show that we have a heterogeneous group of patients that we started off with and have tracked over time. And we're very happy to say that our phase 1/2 interim safety data has not shown any SAEs or serious adverse events. Any adverse events of special interest, these are events that are of special interest to us as a company in relation to what's been described with gene therapy, either preclinically or clinically in studies, which include central or peripheral neurotoxicity, drug-induced liver injury, thrombocytopenia, which is low platelets. We also have not seen cases in our phase 1/2 study of myocarditis or myositis. Itis means inflammation, myo means muscle, and card means heart. So myocarditis is heart muscle inflammation, and myositis is muscle inflammation. The most common drug-related adverse events are nausea, vomiting, and fatigue that all resolve over time, typically within the first 14 days of treatment. So overall, RGX-202 has been well tolerated in the phase 1/2 patients at both dose levels with no SAEs or serious adverse events or adverse events of special interest. So here I'll talk about the biomarkers. Again, I'll focus more on dose level 2. And what we do is we look at we do get muscle biopsies. There's a baseline muscle biopsy and a follow-up muscle biopsy at 12 weeks. We compare the baseline to the follow-up, and the difference is what is shown here. So what we see is at 12 weeks or 3 months after dosing, there's really good microdystrophin expression. So here in the 1 to 3-year-old age group, you see 120% on average. In the 4 to 7-year-old age group, 54.3%, and the 8 to 11-year-old age group, 39.7%. The vector copies per nucleus is how much drug actually can be quantified from the transgene. So from the RGX-202, and that's listed here. And the percent positive fibers refers to the image here on the right. So in a child with Duchenne, this image would be completely dark. You would not see any rimming of the muscle fiber with immunofluorescence. And immunofluorescence is you tag an antibody to the protein, the microdystrophin, and you basically see if it lights up. So classically, before we had such easy access to genetic testing, we would do the immunohistochemistry or immunofluorescence on muscle tissues and see if there's any dystrophin staining on immunohistochemistry. This is a child here that is post-receiving gene therapy, and you could see that the antibody is picking up on the protein that's rimming the muscle fibers, these polygonal shapes here of various sizes, pretty consistently throughout. So we're very happy with not only that we're able to quantify microdystrophin within the tissue, but also that we're able to show that it's localizing itself to the correct place. So I'll go through the functional data. So just to kind of prepare you for what you're about to see, this is our data, and we have the actual data, like change from baseline, and then we compare it to natural history controls. So the natural history controls are extracted from 402 patients from natural history data sets, and we match our patients' baselines to a certain number of patients, whatever's available, to their baseline, and then make sure that they have a follow-up at 9 months or 12 months, which is the data sets that we're going to be showing you. And those patients are then compared in terms of like how are they performing 9 months or 12 months after baseline, and then we compare them to how our patients are performing in the study 9 or 12 months after baseline. So in a cohort of patients that we're about to show you that are 5.8 to 12.1 years at dosing, in naturally progressing Duchenne, as you can see here in the figure, in reference to the North Star Ambulatory Assessment, which is a good surrogate for looking at how the disease progresses, you may see kids improve up until around age 6, but then after age 6, things start to decline. And then sometimes that decline becomes even more rapid. So we're really going to be showing you data not only of how our kids change from baseline, but how they may be improving as compared to other kids who will be in the decline phase because they're older patients that are getting dosed as well. So here are our dose level 2 patients, and improvement is the arrow going up. So the blue bar that you can see is the patient's average and how much they improved compared to baseline. The gray bar is what you would expect to happen after 9 months of time. And we have 65 patients here, for example, in the time function tests to compare. So for the time to stand, you'll see this delta, which is the change from what's expected based on natural history. The delta is negative 1.7 seconds. Now, what you see in the blue bar is like it's gone up about 0.6 or 0.65 or so seconds. If you look at the y-axis here, and that's what they are based on their baseline compared to their baseline. But obviously every child's baseline varies, but you could see that standard deviation bar here that shows the range of improvement that we've seen in the patients, and also the range of decline that you would expect from the natural history. But I'm going to talk to you about the deltas compared to the natural history, but you can see overall that all of the blue bars are moving upward. So every child, as opposed to plateau or decline, is improving in the time function tests and in the North Star Ambulatory Assessment. So for the time function tests of time to stand compared to the natural history, we have an improvement of 1.7 seconds. For the 10-meter walk run, we have an improvement of 0.9 seconds. For the time to climb, we have an improvement of 1.3 seconds. And the North Star, which is a group of tests that can be graded according to your function, that's been well established and tried and true, and is used through a lot of studies, is here also showing an improvement compared to baseline. and a 4.8-point improvement compared to what you would expect in natural history at 9 months. And this is the same data at 12 months, but obviously there's more patients with data for comparison, so we can kind of report this out that at 12 months, we continue to see sustained improvement in our patients, and the time to stand improved by 1.86 seconds compared to natural history, 10-meter walk run improved by 1.1 second compared to natural history, time to climb improved by 1.2 seconds compared to natural history, and the North Star improved by 6.8 points compared to natural history. So again, we're very reassured by this data and see very promising signals for us, not only from a safety standpoint, but also from a functional benefit. And in the US, the FDA did have an approval of Elevidys, and they used something called minimally clinically important difference. And minimally clinically important difference can only be established at 12 months. These are some set values. So you can see this MCID in the gray bars here, and you could see that how our drug is performing compared to that. So obviously we're outperforming the MCID for the patients that we have at 12 months with dose level 2. And that's in the time to stand, the 10-meter walk, and the time to climb 4 stairs. Now, we also we do find that the functional outcomes are important, and these are quantifiable. But what really has a lot of meaning for us as well, and I think also to regulatory bodies, is how does this functional improvement translate to meaning for patients and their families? So we do patient-reported outcome measures. We use something called the POTS scale. And here you could see on the y-axis the scale score mean change. And there's a reference here to what is projected, not from our natural history data set, but referenced in the study here on the paper from Craig McDonald in 2010. And you can see in the blue bars how all the families after a year of being on RGX-202 are showing improvements in these 3 domains of transfer and basic mobility, sports and physical functioning, and global function. So caregivers reported improved function in the home and community as measured by key dimensions of the POTS at 12 months compared to expected decline in natural history. So our interim summary is that AFFINITY Duchenne is well tolerated in 13 patients across both dose levels with no serious adverse events or adverse events of special interest. We have a differentiated therapeutic approach, which may contribute to a positive safety profile. This is a nod to our immune modulation regimen. Our biomarker data support consistent, robust expression, transduction, and sarcolemmal localization of RGX-202 microdystrophin. And we have new functional data to support consistent evidence of RGX-202 altering the trajectory of disease, showing boys treated with dose level 2 demonstrated clinically meaningful improvement in function at 9 and 12 months, exceeded natural history external controls and MCID benchmarks, and showed improved function in the home and community as reported by caregivers. So I'm going to briefly talk about our phase 3 trial design. So a phase 3 study is mainly obviously we continue to monitor safety in the patients, but the primary focus is, okay, you've established safety, but does this actually work functionally? So we modified the regimen a bit in terms of who we're including in the study. So now we have no upper limit of age. We're enrolling boys ages 1 and older. We've actually expanded our criteria for the mutations. So we only exclude at this point, and hopefully this will change in the future, but not yet at this time. But the only exclusion we have, meaning to be more inclusive, but the only exclusion we have at this time is we take all mutations except those children with deletions or point mutations in exons 8, 9, and or 10. So duplications are allowed in 8, 9, or 10, but just not deletions or point mutations. The children should have no pre-existing antibodies to gene therapy. The outcome measures for the 1 to less than 4-year-old patients that are needed to be assessed prior to inclusion in the study are listed and unchanged, except for the 4 and older patients who need a baseline NSAA of greater than or equal to 16. The immune modulation regimen has stayed the same. The trial endpoints are a bit different. So the primary endpoint is the proportion of patients with greater than or equal to 10% of RGX-202 microdystrophin. So what we want to show is that across the board and all the patients will show at least 10% of RGX-202 microdystrophin, showing that we're not going to have any patients that don't have any RGX-202 in their muscle. and that it's getting there, and it's shown diffusely throughout. And the secondary endpoints are timed function tests. So this includes velocity of time to stand, North Star ambulatory assessment, Peabody developmental motor scales, SBN95C for 1 to less than 4-year-old patients, and safety data. And the exploratory endpoints for the 4 and older are SB95C and cardiac and skeletal muscle MRI. Here are the locations for our study sites for the phase 3 study. So right now, this is what we have in the United States. And we also have some sites opening up in Canada. So it's nice to know for the United States, but I'll show you what's going on in Canada. And at this time, we're very close to getting Dr. Campbell's site up and running. We hope to have, I think, 3 to 4 more sites in Canada up and running very soon. But Dr. Campbell, he's in London, Ontario, at the Children's Hospital London Health Science Center. So stay tuned for the new sites to be opened. We're really excited and we're very invested in Canada. Affinity Duchenne, just to kind of give you an overview of the phase 3 pivotal trial. The phase 3 portion of the Affinity Duchenne study is an open label trial. So again, just to hammer this home, for the phase 3 study, we are not having any placebo patients. So anybody coming in the trial is receiving drug. We are expected to submit a biological license application, or BLA, under the accelerated approval, FDA's accelerated approval pathway in mid-2026. And if you need more information, or if you have any questions that you'd like to discover, or you'd like to ask us about whether or not your child may be eligible for the study, you can go to www.regenxbiodmdtrials.com, or you can email our team at duchenne@regenxbio.com. And again, acknowledgements-wise, we're a very dedicated company to patients and families with Duchenne. Well, obviously I'm biased because I'm working on the Duchenne program, but to all the patients that we treat as a company, obviously, but we have a real soft spot, obviously, in the 202 program for treating patients with Duchenne, and also to all the patients in our Affinity Duchenne and Affinity Beyond study for advancing our program forward and teaching us so much, and hopefully paving the path for more kids to being treated.
Thank you so much for all that information. I'm sorry, am I interrupting? Did you want to go through this?
No, this is all good. This is just a nice to know repeat QR codes if anybody wants them.
Well, I have a question, if you don't mind me stepping up with the first question. I'm curious about, with only one site currently almost active in Canada, if a person lived closer to the United States versus London, Ontario, are they able to go to a site there?
Yes, for sure.
Okay.
So these are all the sites that are open in the US right now. So we have California, Colorado, Texas, Arkansas, Virginia, and Illinois. And we're getting a lot more up and running as well. We are really going at light speed. So hopefully you'll see a lot more of these bubbles pop up in the US and also in Canada very quickly.
That's great. Now, a question, logistically, if somebody was to go to the US to be treated and a site opened in Canada nearer to their home, are they able to switch the follow-ups to the Canadian site?
It's very difficult to do that.
Okay.
Not the ideal situation, just because tracking the data, we understand if there's a super need to do that, but I wouldn't enter the study with that intention in mind. I would say but I would also say that the most intense follow-up is in the first 3 months, and we support travel. You can, if you're coming from Canada, probably I would say I would budget 2 months of your time to be near the site because of all the repeat lab work, which could happen regardless of where you are in Canada, right? Because Canada is such a huge country. So, budget about 2 months of your time, then you could fly back and forth. And then after that, your visits become less and less. And then it becomes more like clinic visits over time. So after about 12 months, you end up coming in every 6 months. So it's not too bad.
Now, somebody in the Q&A is asking, what does a follow-up plan look like? I think that you've already addressed that. Now, from my own curiosity, though, in those first 2 months, how often are you going to clinic? Or would a patient be going to clinic, I should say?
Yep. So basically, in the first 2 months, you have some screening procedures that happen within the first 60 days. I would say that's probably between 3 to 5 visits, depending on your schedule and how much time you have at the site and whether or not the child is cooperative for long periods of time. Like some kids can only do so much of screening. And then after that, you have your first dose, your dosing day. And then you usually follow up like every other day for the first 2 weeks or so. And then after that, it becomes like every 4 days, then weekly between weeks 8. Actually, it's bi-weekly between week 8 and week 12. And then it goes monthly until month 6. And then every 3 months until month 12, and then every 6 months after that.
Great. So we have a couple of questions. One is, my son tested positive for AAV9 antibodies. Would he be ineligible?
No, because that's a different viral vector. So you have to just get tested for the AAV8 antibodies to be sure if you're ineligible.
Great. Another person asked, can the children go back to school post-infusion or would they have to quarantine?
That's a great question. So what I would say is sometimes it also depends on the time of year. So if it's really heavy flu season and whatnot, and you want to be extra cautious, you could go back to school, but you have to practice good hand washing techniques. And maybe I would suggest wearing a mask because you're on the immune suppression, the eculizumab, the sirolimus, and the prednisone. So you don't want to get exposed to anything, especially in those first 3 months or so. But after that, I think you're good. You know, we've had kids travel and they've been fine after that. It's just that critical first 2 months was when you're on the max dose. The 3rd month is when you're weaning. So I would probably just recommend wearing a mask if you're at school. And because of the very frequent follow-ups, I would say probably budget about 2 weeks off from school or being able to do work away from school for those 2 weeks.
That's great. Thank you.
Mm-hmm.
Those are all the questions we have. If anyone has any additional ones, we'll just give you a minute or two to get that into the Q&A. This has been very helpful and very informative.
Oh, sure. Great.
I'm looking forward to hearing about more places opening up.
And we're working on that. So yeah, we expect additional Canadian sites to open up soon.
Wonderful.
We just can't
Please let us
announce them
know
just yet.
and we'll make sure to put that out to everybody. Do we know roughly when London will be recruiting?
Let me see. I would say hopefully very soon. I would say like I don't want to overspeak. Don't hold me to this, but at least at least by the end of the summer, I would say.
Good to know. Will you be opening sites in the UK?
Oh, okay. I just clarified September.
There we go. Okay. We'll revise the video. We have the other question. Will you be opening sites in the UK?
Yes, but that's farther away. That's going to take some time still. Yep.
Sounds good. Is deflazacort allowed?
Yes.
That's a good question.
That is.
Anyone else? Does anyone else have anything they'd like to ask the RegenX Bio team? There's another. What about vamorolone?
Yes, but we have an adjusted regimen for those on vamorolone.
Good to
So
know.
the safety of giving vamorolone at the same time as prednisone, which we give for the additional immune suppression, hasn't yet been established. So what we do is we ask that you convert your vamorolone to prednisone equivalent dosing while you're receiving this additional prednisone, and then you can go back to vamorolone because your baseline assessments will be on vamorolone and your follow-up assessments will be on vamorolone.
One of the things that we've had in Canada is the wording is important on the protocol here. And if it says that it has to be an approved treatment, we don't currently have approved treatments. So I'm flagging that for you now just to make sure that the wording allows the same so that no one has to go off their deflazacort or vamorolone. Any other questions, anybody? Every time I've said that, something else has popped up.
No, it's all right.
It's looking like it might be wrapping up. Going
Yeah,
on.
and we're very welcome to receiving any questions. feedback, anything that comes up, just let us know.
Wonderful. And if you can keep us up to date about when other sites open up, we will get that out to everyone right away.
Thank you.
So
100%, yeah.
you can hit, excuse me, you can hit the ground running. We do have one more question. Is travel from the UK feasible?
Yes. Yep. So we just have to work with certain sites because each site has different policies about international patients and like their capabilities of taking the patient at the time, but we will support travel to get into the study.
Wonderful. That's great to know.
Mhm.
And great questions, everybody. Giving it one more second just to see if another one pops up. There's a thank you.
It's a pleasure.
Thank you so much for all this information and for taking
Yeah.
time to give us this information. And
Thank you, Judy.
we look forward to hearing more on your next update.
Hey, awesome.
Stay in touch.
That's great. Thank you so much for having us.
Thank you so much. Have a great evening.
All right, take care. You too.
Thanks, everyone.
everybody.
Bye-bye.
Bye.